we go to the next session, which is on inquiring the molecular tumor characteristics. And what I didn't say, and Dr. Liu and I discussed in the break, of course, we had results on screening, trying to get tissue diagnosis, but there is a feeling within a few years we can do maybe certainly liquid biopsy and molecular diagnosis, which will certainly assist the workup of patients with nodules too. So the first speaker is Dr. Govindan. He comes from St. Louis, from Washington University. He's been key person at TCGA on lung cancer, and I'm very looking forward to your talk. Thank you.
Thank you, Rolf. Thank you, Rolf, for the invitation and your wonderful hospitality. It's a great pleasure to be here. And I'm just going to speak about the molecular changes in lung cancer. And before I talk about the evolution of cancer, lung cancer, I just thought we'll talk a little bit about what we have learned over the past several years. And I just want to bring everybody up to speed. So in the first part, we'll talk about the genomic alterations. Second part, we'll talk about how they evolve. And this is actually what is called the Circos plot. And I'm sure many of you are familiar with this. What this means is that this is a pictorial representation of what is happening in a single adenocarcinoma specimen by doing the whole genome sequencing, which is basically a way to look for all the 20,000 genes, the coding and non-coding regions. And this is very easy to follow if I explain to you in a minute, even if you're not familiar with that. These are chromosome numbers here, as you can see here, 7, 8, 9, so on. And all the letters that you cannot read are single nucleotide variations within each of these chromosomes. And these are copy number alterations and that are depicted here. These are rearrangements within the chromosome, and these are rearrangements between the chromosome. What it means is that a fragment of chromosome 7 is now combined with the chromosome here, chromosome 14. So that's basically how you read this. This particular patient actually is a patient of mine, had over 50,000 mutations in a tumor, and there are only 20,000 genes, but a given gene can be hit in multiple places. 50,000 mutations. Granted, most of them are not important. Actually, almost 99.9% are not important in the disease pathogenesis. And in contrast, you look at the Circos plot of this patient, again, lung adenocarcinoma, whole genome sequencing. You can see a very, very calm appearing genomic landscape here. And the big difference is that this patient had significant tobacco smoking, and this patient had no history of tobacco smoking. So the reason I'm showing this is that this background mutation rate that is very common among tobacco-related lung cancer really complicates our discovery of novel genomic alterations. And when you have that much background noise, picking up the signal becomes a challenge. And we have overcome that with a lot of informatic approaches, but I just want to have an appreciation of this. And very quickly, before I go further into what we saw, just a basic introduction of what is whole genome sequencing. You look at all the 20,000 genes, both coding and non-coding regions. And in fact, 8 years ago was the first time, anytime, anywhere in the world, a whole genome sequencing of a cancer patient was done in St. Louis. And after that, now several hundreds of whole genomes have been done. And in fact, Human Genome Project was completed only 15 years ago, which took about 10 years, $3 billion to do one genome. And when we did the whole genome, the first time we did this for just $1 million, from $3 billion to $1 million. Now we can do this for a little around $3,000 to $4,000 for one whole genome. So the cost has come down very dramatically. The advantage of a whole genome is that you get the full thing: coding, non-coding regions, structural rearrangements, copy number alterations, single nucleotide variations, insertions, deletions. Informatically, it's very challenging. It takes a lot of time to analyze the whole genome sequencing. The whole exome, on the other hand, looks at the coding regions, as you can see here. But the disadvantage is you don't get information on the non-coding regions, and rearrangements are hard, but you cannot get information on that. The transcriptome sequencing gives you information on the structural variations like translocations and fusions, inversion, etc., and also point mutations, insertions, deletions, etc., along with gene expression. So we often use exome sequencing and transcriptome sequencing together, the cost is still about 1/5 of what we pay for the whole genome sequencing. So it is really very useful. And I just want to speak a little bit about the genomic alterations from the Cancer Genome Atlas project. Cancer Genome Atlas project was a large National Cancer Institute initiative in the US, where over 10,000 tumors were sequenced. Most of them are whole exome, transcriptome, and some whole genome. And we've learned quite a bit. Lung cancer was a prominent part of the TCGA, and we sequenced over 100 lung adenocarcinomas and 500 lung adenocarcinomas and over 500 squamous cell carcinomas. And there were 3 papers published out of this, the primary papers in 2012, 14, and the most recent one is 16. And I just want to thank Rolf and Solange for their help, and the ETOP investigators and other institutions in this wonderful land of Switzerland, and without whose help this project couldn't have been completed within the time. And what have we learned? In squamous cell lung cancer, we found that the oxidative stress response pathway is commonly altered along with pathways involved in squamous differentiation, and the common alterations involve SOX2, Notch1 and 2, and also expressions of FGFR along with mutations in NFE2, L2, and KEAP1. And for the first time, we found that nearly 2/3 of squamous cell lung cancer patients potentially have an actionable alteration that we could be going after with therapeutic approaches, and several studies are currently ongoing to sort that out. Compared to what we had known about lung adenocarcinoma nearly 15, 20 years ago, now we know a lot more about this, and we found that I can simply summarize in one sentence what is lung adenocarcinoma is. It is altered by mutations or alterations in the receptor tyrosine kinase pathway, one way or the other, and various members of the receptor tyrosine kinase pathway. And these are the distribution. It is all outlined in the TCGA paper. And by doing this whole exome and transcriptome analysis, we did find, even with 230 samples, as we published in 2014 in the Nature paper, 2/3 of them, or actually 3/4 of them, had a potentially actionable alteration. And if we had drawn the pie chart 10, 15 years ago, this would have been very empty, except for about 20% of KRAS mutations. So in a span of 10 years, we have now discovered alterations in nearly everyone except about 20% of lung adenocarcinoma. And that was in 2014. We actually went further in the last 2 years, and drilled down into those what we call oncogen negative group, and we actually did further analysis, and we found more alterations in the receptor tyrosine kinase pathway, an additional 13%. So I can say now, today, that we do not know what's driving this cancer in about only 15 or 20% at the most, and we have figured this out in 80% of those patients. What are the key drivers? And that's only the very first pass at this. We got to understand a lot more as to what's driving this cancer, but the key drivers and that are potentially causing these cancers are identified in nearly 80% of lung adenocarcinomas. And if you notice, all of them involve the receptor tyrosine kinase pathway in various forms, and this is very nicely outlined in the paper. We are beginning to integrate not only doing the DNA, RNA sequencing, and methylation assays to really figure out what are the various ways these pathways can be altered. The content of the slides are not that important. The reason I put that is, by integrating this analysis, we do find in some instances, for example, the KRAS mutation is not seen at the DNA level, but the evidence of that pathway is activated at the proteomic level. So we really, by integrating this, we find these kinds of alterations not picked up by one, but picked up by others. And based on that, we can group them in various bins. And we're really trying to understand the biology of these cancer as we go along. And it's really powerful to integrate these tools. And by actually doing this, one thing we really found is that lung squamous cell cancer is a very different lung cancer than lung adenocarcinoma. You know, the only thing that's common is that they arise in the lung as an organ, but they are caused by alterations in different pathways, alterations perhaps in different cells. For example, lung adenocarcinoma is probably driven by mutations involving the type 2 alveolar cell that is important for lung alveolar regeneration. whereas lung squamous cell cancer is driven by alterations in the basal cell. And what is interesting is that these cells can go to and fro. For example, small cell lung cancer is almost always caused by mutations or loss of function in both RB, retinoblastoma gene, and P53. If there are no mutations in both RB and P53, there is no small cell cancer. You knock these two genes in animals, you get small cell lung cancer. And that happens predominantly in the neuroendocrine cells to get small cell lung cancer. But potentially a type 2 alveolar cell could possibly have an EGFR mutation, become a cancer, and lost P53 along the way. And then during the course of treatment, somewhere along the line, it loses RB. Now it goes down the small cell pathway. And that explains why some patients with lung adenocarcinoma, EGFR mutant lung cancer, upon getting EGFR TK inhibitors, now suddenly become small cell lung cancer, because these cells are very plastic. These programs are very plastic. I'll remind you once again that you can take an adult skin cell and then turn off just 4 genes, turn on 4 genes, and you can create an embryonic stem cell and this adult stem cell. And so these pluripotent cells are integrated because of, are caused by these alterations. And so it's really remarkable that we can see these things. And these are unique alterations, as you see here. And you have this, some are shared, but then most of them are specific for lung squamous cell, specific for lung adenocarcinomas, and there are some that are shared, but they're very distinctive. And we asked a question, how common lung squamous cell cancer to lung adenocarcinoma in the Cancer Genome Atlas project? It turns out that the lung adenocarcinoma is not similar to lung squamous cell cancer. In fact, the lung squamous cell cancer is more similar to head and neck squamous cell cancer than lung adenocarcinoma. So that's actually is an important observation, that squamous lung cancer has more in common with squamous head and neck cancer than lung adenocarcinoma. So that's important. Either way you look at it, mutated genes, amplifications, or alterations. And then more importantly, with all the mutations I showed you, what it means is that these tumors have very high level of mutation burden. In fact, the average mutation that we see in the lung cancer, squamous or adeno related to tobacco, is about 200 tumor mutations per tumor on average, if you look at the coding regions and non-synonymous mutations. And that's about 10 times higher than what we see with acute myeloid leukemia, 10 times. And in fact, the lung squamous cell cancer and lung adenocarcinoma are one of the tumors with the highest mutation, with only exception is melanoma. What's common between all three things? They're all caused by carcinogen exposure. So this constant tobacco smoking causes these alterations over the lifetime of a cell, and these cells pick up these mutations, and eventually one of them causes the disease and causes transformation. But these mutations precede even the development of cancer, and then they just hitchhike along with the rest of these things. So that's why we see a very high mutation burden. What this means is that this has a potential to cause some neoepitopes. These are alterations that can cause some antigenic recognition by the immune cells, and these are called neoepitopes. And we find that, in fact, if you look at least a lung cancer and ask how many of these lung cancer tumors have at least 5 neoepitopes per tumor, the answer is more than 70%. What does this mean? This means that lung cancer is a very rich tumor for creating personalized vaccine based on the neoepitope alteration. I'll get to that towards the end of the talk. So that's the first part of the talk. What have we learned from all these things? And what do we have to learn as we go along? I would say that the key thing, very immediate priority for us, is to find the rare mutations. I told you that nearly 80% of lung adenocarcinoma, we know what's driving this cancer. What about the next 20%? That's still a very large number, considering how common lung cancer is. Second, we really know a lot about the DNA alteration in lung cancer or in many cancers today, but we really have not understood the RNA alterations or how the genes are upregulated or overexpressed. They themselves can cause cancer, not just the DNA sequence alterations. And we've really not done a very good job of that, so we should work on that. And the third, the epigenetic alterations that happen in these tumors are very profound. So how can we really understand these things? And that's something we are going to be studying as we move along. And then more importantly, it's not enough to say that we have found EGFR mutation in lung cancer. And this EGFR mutation does not happen in a vacuum, and they happen along with several other alterations. And we have only a very limited understanding of what else is happening in these tumors. And what else happens after therapy, because because after all, even if you have a spectacular response to EGFR tyrosine kinase inhibitors, very soon these patients develop resistance and then they become very refractory to therapy. So what's really guiding these alterations? Is there a theme that we can foresee and we can develop treatment that we can prevent the development of these alterations? And so these are very important. And so to answer the first three questions, finding the rare alterations and studying more, there is going to be a massive effort currently ongoing based on the power calculations we did. It turns out that given the high mutation burden we see in lung cancer, to pick up the signal from the noise, we have to sequence over 10,000 tumors, lung adenocarcinoma, and that's the project we just started. And the very first step of that will be what is called the Cancer Driver Discovery Project, where we'll be sequencing over 3,000 lung adenocarcinoma. And I've been talking to our colleagues here, and hopefully with your help, we'll complete this project in a fairly timely manner. And this time we are going to do whole genome as well, apart from whole exome, in order to understand coding and non-coding alterations and really looking at all the changes. Technology has improved. We can do this in paraffin, not perfect, but then we probably will try to get as much fresh tumor as possible, and then we'll do that. The second part of that effort will come from the ongoing prospective study in the US called Alchemist trial, which will screen over 8,000 patients. Already 1,500 patients have been screened. And part of that study involves using therapy for EGFR mutated ALK positive patients and using immunotherapy as well. So we'll be collecting tumors from all of them. So with all these understanding, we'll probably identify more these rare alterations and do a much better job in understanding the transcriptome alterations. So what about the evolution of this tumor? And in fact, this is a topic by itself, and I would say that this is an entirely a day-long symposium you can have on cancer evolution, and I'll do that in the next 10 minutes that I have to really help understand what is happening in the evolution of the tumor. And you know, the tumors really actually pursue an evolutionary pattern very reminiscent of what happened in the evolution of species. And in fact, the genomic alterations that we see in the tumors are they recapitulate what even Darwin scribbled on the back of a paper on the evolution of the species. And I'm in the Europe, so I can talk about evolution without any hesitation, unlike in the US. So I am completely unrestrained now. And it is really, you can see that in a beautiful way. And you know, just so that you understand, this is a normal cell gets transformed when there's a critical event. For example, in lung cancer, we showed 4 years ago that the EGFR mutation is one such example, and that is present in every progeny of that founder cell, and that is called the founder clone. In other words, you take a tumor, biopsy any region you want, any place you want, you will find that mutation. So cancer is a monoclonal event still, but then what happens is that we appreciate now much better that these monoclones or single clones have now given rise to subclones. So as you notice here, the founder clone has this mutation, for example, here EGFR, and every single progeny has that, but then they picked up various different additional alterations. A subclone here has this green mutation, and just to simplify here, and here is the yellow one, and then there are other subclones, the B1 branches to have different kinds of the third mutation. So that way these things branches, and based on that we can construct the phylogenetic tree, and the length of the tree depends on the burden of mutations, and you will see that frequently. So just to help you understand that, how can we do this? It's a very simple concept. You know, think of this. Mutations happen typically in one of the two alleles, and if there is 100% of these tumor cells have mutations in one copy, so your variant allele frequency will be 50%, and this is a single cluster of mutations, so that talks about one clone. And then when you have this in a smaller number, so the variant allele frequency go down. So using the variant allele frequencies in deep digital sequencing, now we can find 3 clusters, for example, here, a cluster that's about 50%, there's a cluster that's nearly 100%, there's a cluster that's a small number. So you have 3 different clones, and now we are getting better and better informatically to analyze this, and also getting better in looking at this. And so the deeper you go, the greater the chances you're going to find these clones. And if you have a very superficial analysis, you'll come away thinking that there's only one clone. And here is an example. If you do only this in fact, this was considered state of art until 4 years ago, where you do 30x whole genome, 30 times you cover regions of this, or 50x whole exome, and you may find a few clones here. This is the founder clone, these subclones here as they evolve. But then if you do a deeper sequencing where you go 300x whole genome, 400x exome, very deep coverage, pretty expensive, and then you can do targeted genes, nearly 10,000-fold, you find multiple subclones that you would not pick up otherwise, as you can see here. So it's really a complex thing. So you go from 5 driver mutations to 10 driver mutations, and you find not only one cell of origin, but a couple of cell of origin. So it's really, the more you study, the more you find. And you can see some signatures here, various different signatures caused by tobacco smoking based on the nucleotide alterations. And this is something that we see increasingly caused by upper-Beck, an enzyme that is important in viral defense, and that probably causes intrinsic mutagenesis. So different carcinogens leave behind different signatures. The C2A is an extremely well-known mutation signature, and based on that, we can really figure out what are those mutations acquired early or caused by tobacco, what came on later, and with upper-Beck. And the upper-Beck enzyme was recognized about 15 years ago simultaneously, both to contribute the mutagenesis in the cell and also for viral defense. And we see that pattern, particularly the upper-Beck enzyme levels are much higher in some cancers, lung adenocarcinoma and squamous cell lung cancer being one of them. So as the tumors evolve, various events contribute to that. Early on, it's probably caused by tobacco smoking relevant to lung cancer. They cause certain specific alterations in specific genes. And as the tumors evolve, subsequently the local intrinsic cellular processes pick up, and one of them is the upper-Beck enzyme, and that causes additional alterations in key genes, and that lead to further evolution of this cancer. And it's also becoming clear that whatever germline alterations you have or the patients have, and that can actually predispose to somatic alterations. And I don't have time to go through this in great detail, but it turns out that patients who have certain germline alterations are at high risk for lung cancer. And also, if you have a certain germline alterations, you don't get some somatic alterations. For example, germline ATM, an important DNA repair, and the P53 mutations in the tumor are not seen concurrently. So clearly the tumor alterations can be sculpted by some of the germline susceptibilities that people can inherit. And when we look at this, it turns out that literally every tumor that we see has more than one clone, and they have significant alterations. Take, for example, breast cancer, renal cell cancer, lung cancer, name it. They have multiple different clones, and literally these things are not only variations between the patients, but within the tumor, as Charlie Swanton nicely showed in this publication, that within the tumor there are regions that are unique. And a simple analysis by taking a tumor, cutting in multiple sections, sequencing each of them, he asked a very simple question. How common are these mutations that are present in every single section? Take a tumor now, you cut into 4 sections. What proportion of these mutations are present in every single fraction? The answer is about 50%. And others are private. And that's an important message from these tumors, from these studies. And about half of them are shared in every single segment, and the other half are very private, present in one corner of a tumor. We are talking about one tumor that the surgeon, like Walter, cuts it out, and then you just take one tumor. Only half of them are present in every single section. So it's really, really interesting. Small cell, on the other hand, is not as complex as non-small cell in terms of clonal diversity, which may be a bit of a news to you, but we'll get to that at some point. And most of the heterogeneity studies have focused on DNA alterations, but we see that in the RNA Seq as well. For example, glioblastoma, there are very different subtypes, and that's between the patients, and that is used for treatment today. But then if you take a glioblastoma, cut this into multiple sections, do the RNA Seq analysis, within the tumor you have various different subtypes in one region versus another, showing how complex these things are. And these variations actually may alter the way we think. So what does this all mean? I have only about 5 minutes, so I'll try to summarize as much as possible. And one of the things that came up in the early studies is that early mutations are caused by tobacco-related events. The later mutations are caused by upper-Beck. And this actually neatly explains why half of our patients with lung cancer get lung cancer, even though they quit smoking 15 years ago. And what probably happens in these patients is that these are the early mutations that sets the process. patient stops tobacco smoking, but still, once the process is set on, the intrinsic ApoBec-related mutagenesis takes over, and that completes the transformation. So that explains why some of these patients still get lung cancer even though they quit smoking 10, 15 years ago. Why is that important? ApoBec is targetable. So potentially, if you can identify these patients earlier on, and you could probably prevent the cancer, and chemo prevention may come through these approaches rather than giving something in a brute force. Can we use this as a biomarker? Two things I want to tell you. If you take a single region, you can actually biopsy, you can do the genomic analysis, come away thinking that something is clonal, but then you realize that if you sample this area, you will think these are clonal events, but then if you do multiple events, you find that whatever that you find here is not exactly clonal, it's a subclonal event. So unless you do multiple regions, you really get a false illusion that it is clonal. And it also turns out that the tumors that are really complex may not do well. There is something called just the right level of genomic instability. Too little, the cells cannot adapt, they die. So they don't really cause problems, and patients may do very well. Too much of genomic instability, the cells that become it's catastrophic for the cells, and that may be actually good for the patients too. So the cancer cells have to find the right amount of genomic instability, not too low, not too high to survive. And when we look at the prognostic models, we commonly make a mistake looking at the high versus low, but it may be that the too low, too high, both may do well. And there is evidence now in lung cancer, also in colorectal cancer. And the right in the middle is actually good for the cancer cell, not for the patient. So that's something we have to be very sophisticated in our thinking, and that's something we'll be studying as we go along. And the other reason why it's important is that the tumors metastasize, and clearly metastasis is governed by heterogeneity, and it's possible that a primary tumor may have a clone that can grow preferentially in the brain, some may grow in the bone, some may grow in the liver. And we and others are doing studies looking at the brain metastasis, seeing what is guiding them. And the important message from this work is that the brain tumors can be driven by a clone that is not so apparent in the primary tumor. In other words, eventually, we may be looking at either the CSF or cell-free DNA analysis or brain biopsy to really guide the treatment, because you may not be able to detect what is driving the brain tumor by looking at the primary tumor, partly because of what I said, the illusion of clonality. You may not pick up those clones that grow preferentially in the brain. And so these are important why the tumor cells evolution is really critical in our thinking. And that's all what we call the spatial heterogeneity. And what is important is that over time, these tumors acquire resistance, and that also can guide therapy. For example, here is a tumor that has actually multiple concurrent alterations that prevent the tumor from responding to BRAF inhibition. And KRAS tumors, they're not just driven by KRAS mutation alone. The context matters. Some of them actually have loss of CDKN2A, some of them have loss of LKB1 or STK11, and some have P53 mutation loss. And these three subgroups may have to be treated very independently because of the heterogeneity within the KRAS mutant tumors, and they seem to behave a bit differently. And so that's something we need to sort out as we go along. I won't go into the detail, but this was discovered very nicely by the John Haymack paper on cancer discovery in 2015. So the important message is that the landscape of these concurrent alterations matter, and we cannot treat them all in one group. And the same, I think, will be obvious in the coming years in EGFR mutation. The presence of concurrent alterations may dictate the response to therapy and also clonal evolution. And now we have evidence that in acute leukemia, the response to decitabine epigenetic therapy is dictated by the presence or absence of P53 mutations. Those who have a P53 mutation tend to do better than those who don't have P53 mutation. And also the neoantigens, those tumors that respond to PD-L1 blockade actually have a high mutation burden, and they have clonal neoantigens, and they are not subclonal neoantigens. Those who have subclonal neoantigens or low mutation burden do not do well with checkpoint inhibitors, and that makes a lot of sense. If you have a very potentially targetable mutation, once you release the inhibition on the T cells, those patients do pretty well, especially if they are in the clonal event. And then over time, this is the temporal evolution of heterogeneity. And this is the work done by my colleague Tim Lay, where they showed that in leukemia, patients present with these multiple clones. This is the founder clone. At the time of disease presentation, they have 4 or 5 different clones. Treatment eliminates most of them. But then at the time of disease relapse, one particular clone dominates it, and that kills the patient. So the serial tracking of the evolution, the mechanisms underlying the resistance, can only be understood if you study this better, and that's going to be very important as we study more and more. And that's happening increasingly in lung cancer as well. We don't have as much data today to talk about in terms of evolution over time, but we are already beginning to see that in lung cancer and various other tumors as well. 2 well-known examples. Since it's too well-known, I'm not going to talk about this. I'm just going to mention it in passing. EGFR mutant tumors do not respond to therapy after a while because of emergence of T790M mutation. Why that happens is not so clear, and we are trying to understand the genetic basis of that. And we have multiple therapies. This is a patient of mine who had an EGFR mutation, responded very well to EGFR inhibitor, developed progressive resistance, and then now responded beautifully to the T790M inhibitor. So really we can affect them in terms of therapy once you understand them. So we have to not only do serial biopsies to guide therapy these days, but it's very critical for us to understand the mechanisms of resistance and the tumor evolution over time. This is an extremely great example of how immune therapy also will be sculpted by tumor evolution. Here is an example of a patient with a KRAS mutation got immunotherapy. And this is a patient who responded very nicely with immunotherapy that targeted KRAS mutation. And it turns out that one of those lesions was progressing, and biopsy of this lesion showed there's a loss of chromosome 6 allele. Why is that important? That's why the HLA molecule C082 was there, it's HLA-C, and that is important for the KRAS neoepitope presentation. So the tumor cells very smartly delete the HLA, particular HLA, and if that is not there, even the mutant thing cannot be presented, and the immune cells cannot see that. And this is a very neat mechanism as to why this lesion did not respond to immunotherapy, because it lost of the HLA allele. And we see that in tumors, lung cancer, actually fairly often. And finally, in the last minute, I want to actually leave you with a more optimistic point. And this is the Achilles heel of tumor: genomic instability, high mutation burden. We can exploit this in 2 ways. One is by using DNA repair inhibitors like PARP inhibitors, you may be able to achieve response to therapy because these tumors are genomically unstable, somewhat like BRCA deficient tumors. Second is they are rich sources for vaccine therapy. So today we are launching studies where we're going to sequence a tumor. We find what are altered. We do the neoepitope prediction using computer algorithms. And we actually do RNA sequencing to ensure that they are expressed. And then we take the sequence and create them artificially, take the patient's T cell, train them, and then infuse them back. And so that these T cells are trained to look for the new antigens. And then we are going to add additional checkpoint inhibitors to release the brake in order to augment therapy. We already treated 8 patients with melanoma. We showed that nearly 70% we can really generate antibodies for the predicted neoepitopes. And with all these things, I think using the genomic instability as a vulnerability and eventually using these alterations and using a neoepitope-based vaccine, I really think we can make substantial progress. These treatments are complex, but cancer is complex, and the genomic alterations are very profound, and we have to approach it very sensibly. And in the coming years, I really think we'll make substantial progress. And at this point, we have learned that the lung cancer is a complex disease, and the mutation burden is much higher, but we can use that to our advantage by doing very important studies over time. And I want to stop here and thank you for your attention.